So I'm Mike Tobin. I think I'm giving the first presentation here. So I'm used to speaking to undergraduate students, so I am used to people falling asleep in the first lecture. So I will expect that. So what I'm going to talk about today is about what multivariate analysis might be able to be used for in the pharmaceutical industry. So that's a topic I've come to over the past few years. There we go. So I trained as a pharmacist, and then I worked teaching undergraduates in pharmacy, and then I worked for a small pharmaceutical company, and now I work for a large pharmaceutical company. And what I've done during most of that period is look at material science. How do you make a powder make a better tablet, and how do you make a powder make a better inhaler? However, I now work for an organization. We think we generate 6 petabytes of data every year. So that's an estimate that we got from our IT people, that the amount of extra storage that they buy every year, hard storage and cloud storage, is about 6 petabytes of data a year from one organization. And that data is relatively structured. So we might be able to do something with it. This is what I do in the rest of my time. These are my children in Berlin. I have 4 children. So the 3 children were very excited to be in Berlin to see the panda. They'd never seen a panda before. The 4th one, he was very excited to be in Berlin because the drinking age is lower. So, but that's what else we do. Okay. So if you're a pharmaceutical materials scientist, so that is the world's most perfect sphere. So that guy in that picture has spent his entire career trying to manufacture the world's most perfect sphere. It's the most perfect round thing on Earth. So all you need to know about that thing to understand it is you have to know what it's made of, and then you have to know what its radius or diameter is. You can work that out. Everything else you can infer from that. You can work out how far you could throw it, how far it would take to hit the ground, almost anything, just by knowing 1 or 2 parameters. So if something's easy to measure, data analysis is relatively easy. Sometimes, but then actually the particles that we look at are not really simple, perfect spheres. So on the left-hand side, there's a huge crystal that I found in Vienna this summer. And then on the right, there's a single crystal that we would use to generate a crystal structure. But you can already see that the one on the right is no longer a perfect sphere. And it has been grown specifically to make measurements on. But it's already more complex than we would have wanted it. That's what our APIs actually look like. So there's a series of APIs that we've manufactured over the years, and I would defy anybody to come up with a single measurement which would define any aspect of those particles. So pharmaceuticals material science is a topic where understanding complex data and understanding large amounts of data has driven me towards this topic. So if you've got an API like that, and the thing that it resembles most of all is cousin it, how are you going to characterize it? How are you going to understand it? How are you going to say to your colleagues, I want a better material? So that was the thing that we had a few years ago. You can understand that we've been looking at the outside of particles. We now spend time looking at the inside of particles as well. You can take particles and they can be the same size, but if one of them is like a thin balloon and one of them is like a football and one of them is solid, they're still different. So that's what we spend a lot of our time doing. So we can understand imperfect particles using better data system. I don't know if these people are here. We use this system a lot, the morphology G3, where we can get lots of different measurements on particles, size, shape, size distribution, shape distribution. These all make a difference to how a material performs. Okay? And so we're using that, just taking that data. If you're trying to do continuous manufacturing, you can take the data from that, and it turns out you need size distribution, shape distribution. And if you do that, you can perhaps understand what's going to work in a continuous feeder. So that was the challenge that we had. We've got a lot of complex data. What are we going to do with it? So what I decided to do was I would define what a modeling or analytical system would need in order to work. So you would need, it's able to cope with large amounts of data. G3 files are gigabyte size files. We need a terabyte size backup to take all the data that we've generated from G3 over the years, because it's taken a photograph of 10,000 particles and then taking a load of measurements off it. Whatever analysis that we do has to be transparent and reproducible. So if we do a measurement and build a model from it, somebody else should be able to apply exactly the same techniques and come to the same answer. So they should be transparent and reproducible. If, for some reason, we want to validate the model, that's related to the second one. It has to be transparent. We have to explain to others what we've done and why we've done it. And of course, the data, the system should be capable of evolving. As more data comes in, the model evolves. It would meet any expectations that the regulator has for that evolution, and so you go from there. So what we're really dealing with generally is variability. Actually, even some of those terrible materials that we have in there, we could formulate, as long as the parameters that we put in place to formulate them were the same the next time, rather than if that material suddenly changes and goes from being cousin-it to beautiful spheres, then we will probably have a problem. Okay? So it's variability. And what we're ultimately interested in is robustness. It's the robustness of the system to cope with that variability. So when you're doing manufacturing, it's the variability that you want to capture and understand, not just the raw parameters. We are all in this room, I'm going to assume, everybody that I know in here is good at their job. So you're going to come up with a process which, under the right circumstances, will work. The thing that's going to trip you up is if it changes. It's the variability that we're interested in. The world is multivariate. We saw on the last slide. And so where is that driving me to? So this was a slide I made a couple of years ago. And there's lots of ways and lots of techniques which could conceivably meet all of those parameters that I said of being reproducible and but the one that and I certainly speak to the people, there might be people at this expo that do Bayesian analysis and neural networks, and they will be able to, sometimes they will be the correct techniques for that purpose. But in general, the pharmaceutical industry has adopted multivariate analysis most widely. Okay? So those other techniques are possibly right for your application, but the most widely used ones is that suite of techniques called multivariate analysis. So if you look in the literature, up to 2016, they were being used almost 10 times as much in academic studies as that, and they are very widely used in industry. Those techniques have their place, but the one that's been used a lot is multivariate analysis. So we've gone on and we've tried to capture all of the why multivariate analysis is used so widely and how it's used most widely, and so that's a plug for the book. Okay. So multivariate analysis. So what you do with multivariate analysis is take these large data sets and look in the data sets for where the noise and variability is. And that's essentially what we're trying to capture. What has changed since the last time? My process used to work, now it's not working. What has changed in the interim? It's a powerful suite of techniques that's been used for that purpose. And of course, lots of things might have changed. Some of them have changed in a small way, some of them have changed in a big way, and you have to capture that in some sort of form that we can explain to other people. And how the people that developed that, they said they took a 3D space and projected it onto a 2D space, and that's one way to do it if you're doing PCA. But these techniques were developed in the last century. They gradually moved into some manufacturing industry. John McGregor and people like that moved them into the paper industry and the oil industry. And then over the last 10 to 20 years, they've gained adoption within the pharmaceutical industry, these techniques. And why, going back to why are these techniques so widely used, the evolution of the regulatory framework which would allow us to use them has gone in parallel with that. And as you can say, well, which is the chicken, which is the egg? The people that were writing the guidances for how can you use these things in industry, a lot of them were chemometricians. They were saying, going back to the regulatory approval and things like that. So we all know, and there'll be a lot of talks, and every stand will be aware of this, that there's a series of regulatory expectations that we have when we put something in. The suite of techniques that chemometricians have used, called multivariate analysis, have been put together with those things in mind. So the guidances on model evolution, on QBD, they were written with the expectation that multivariate analysis would be able to fill those regulations. So when you start down the development path, you would think all of that, things that the industry needs, things that the regulators need, when they've written those, they've done that with the suite of multivariate analysis techniques in mind. By the same token, if the people that have developed, the support mechanisms to allow us to use those techniques have in some cases validated their systems. So multivariate analysis can be, and chemometrics can be done using the SIMCA tools. They've been validated, and they meet a whole new suite of expectations, which is 21 CFR 11, which would allow them to be used as decision supporting in manufacturing. So when you use multivariate analysis techniques, you're thinking, if I use them correctly and I use them within the framework, ultimately they could be validated. So it meets all of those expectations. Also, it's not, when you develop a model, you don't then stop because things do evolve. The techniques are very well suited to those things when the model evolves, new information is incorporated, that's been captured in guidances, and multivariate analysis will meet them. So what has MVA been used for, and as far as we're aware of, in the pharmaceutical industry? So it really started in PAT. The chemometricians started using it a lot. So they're looking at drug content, blend uniformity. They use that, they developed the chemometrics, they developed the principal components, and they've used that a lot. And so real-time release methods have used chemometric and multivariate techniques in that. And that's where it really got a foothold, when it started to meet expectations. And now it's very widely used for that purpose. If you're developing a real-time release on a PAT method, you'll probably be using chemometrics and multivariate analysis to get to your endpoint. There perhaps are other ways of getting there, but we're a relatively conservative industry. The fact that other people have already been through the process means that it's a good way to go. So that's been very widely used. We've used it in our material science. So if you've got a new API, this is what we use it for. If we've got a new API, we make some measurements on it. We are not looking at what it's different from. We look at what it's similar to. And so we say, a material with this set of properties tends to be easy to process, difficult to process, impossible to process. We do some modeling to say this new material is similar to what's out there. That's not a validated technique, but we find it very useful internally when we can go to our colleagues and say, this material is horrific. Other applications, small molecule API production, it's used a lot. You know, that can be continuous or batch. This goes back to the PAT and chemometrics and things like that. So it's used a lot to model those materials. Drug product, that's where, you know, when you're making drug product, a lot of that blend uniformity or whatever else, or blending endpoint, real-time release. You know, BMS has publicized that we have a real-time release method for one of our products. It's all based around chemometrics, multivariate analysis. Excipients, there's some excipient people here. If you go to Hall 8 and the excipient people, DFE use this to monitor their product. It's ideal for that. You know, these are large volume materials. They can look for changes. FMC have done it in the past. A number of excipient companies look at their product because it's ideal for that. A lot of excipients are pseudo-continuous manufactured. You know, they're operating all the time, and occasionally they'll bag it up and call it a batch, but they've got a rich data set which allows them to see when the process is drifting. If you've got an almost continuous process and you want to monitor it at 3 a.m. on a Tuesday morning, that's the way to do it. Large molecule, upstream and downstream, I'm less familiar with that, but there's a lot of specialty people that know how to do that. We've looked at it for fermentation, which I think is the upstream process. I'm showing my ignorance now. But fermentation, when to add materials, when the yield is there, you can do that. You can do the downstream stuff and purification. Real-time control of those systems. We haven't got many published applications and filed applications where we've left the model completely in control. But if we move to Industry 4.0 and lights out manufacturing, we'll have to get there. People have got some ideas of how to do that. Maybe I'll be told differently. We don't think people have got there. So when we come up with multivariate analysis, this is what myself and the editors of the book came up with, is you come up with a workflow. But the most important part of the workflow is up there at the top. You have to have a problem statement. You can never just take petabytes or terabytes of data and put it through and see if an answer emerges. You've got to probe it and say, have I got a problem statement? So the problem statement might be, can I monitor the concentration of API in a blend by a particular spectral pattern? Or what has changed in my process to make the system stable or unstable? But it's not a technique for just throwing data at something and seeing if answers emerge. You usually have to go in and probe and say, I've got a specific question. But after that, it should be relatively easy. If you've got data, you have to have data as well. You know, it's a technique for analyzing data. If all you have is your prejudice about what's causing the problem, but no data, multivariate analysis isn't going to help you. So whenever you have a challenge, whenever we have a problem, we start with a problem statement and then see if we've got the data and the techniques to answer it. And that can be applied to all of those things that we said: small molecule, upstream, downstream, real-time analysis. So we've found it to be a very flexible suite of techniques. Okay? Of course, it's not just in R&D and manufacturing. People use these techniques for all sorts of things. This was a trawl that I did through the literature a couple of weeks ago, and you can do it for antibiotic use. You can do it for looking, there are people in our organizations. Bristol Myers-Squibb is a big cancer company looking for trends in how people's cancers develop over time and see if there's any signals that they can measure and go in with. So it's not just restricted to that, but they're being widely used in all sorts of areas wherever you've got data and a problem statement. Okay. So what I wanted to say is that it meets all those criteria that I put out. It's transparent, can be validated, and things like that. When used correctly, so that's not to say that if you use multivariate analysis and you use it incorrectly, the FDA is going to validate your model. You still have to follow a set of rules. You still have to know what you're doing. But if you do that correctly and follow the guidances, it should be accepted. And of course, now there's an ecosystem. There are products which you can use. There are people that know what they're doing. If you advertise for a chemometrician, you might be able to find one. We always have the thing, do we try and teach a chemometrician pharmaceuticals, or do we try and teach pharmaceuticals, do we try and teach chemometrics to somebody that knows about pharmaceuticals? In general, my thought is it's easier to do the first one. I think chemometricians and multivariate, they're born, they're not taught, but you can teach them about the pharmaceutical industry. So there is an ecosystem of people out there that if you had a challenge, there are probably consultants, there's certainly full-time operators you could hire to attack your challenge. Okay? And I think that was where I was going to stop. And just another plug for the book. Thank you very much, everyone.
Hello, good morning, and thank you for the speech.
Thank you.
I wanted to ask you, in the last few years, there was a lot of development in also tools for machine learning and other kind of analysis.
Yeah.
And they are also, from a computational point of view, they are becoming more available because of the power of the computer and whatsoever. Yeah. My question is, on one side, multivariate analysis is very easy to understand. It provides also a good way, an easy way to understand the variable. Yeah. But on the other side, machine learning is coming out, and it's also new methods to understand the meaning of the variable behind are developing. When do you think it will be accepted also in this kind of application in industry from a development point of view?
Well, we were literally discussing that last night. Machine learning techniques are very, very powerful. The question is of whether we know enough for them to be transparent. We can't say the machine's learned something and it can make decisions. So I think the frameworks are not there so that you can meet all the ICH guidelines to do that. Somebody will be thinking, and I think in some time people will get there. But some person will have to develop those strategies and protocols so that it meets all those expectations. I haven't seen yet that the people that would come together to come up with, like equivalent of Q8 or Q12 have done that for machine learning. But all that takes is a will that they do so. So I think in the next few years. Oh.
Don't you think that it's more a problem of culture within the ICH, who create the guidelines?
That's entirely possible that the people that are heavily invested in multivariate analysis might be less enthusiastic about machine learning. But if you have an application for which machine learning is the only solution, and it's the only way that enables your process, then you might be the person that trailblazes. You know, we talk a lot in the pharmaceutical industry. You have to be, everybody wants to be the first to be second. You know, if the first person that takes a machine learning technique and tries to pilot it through, you know, a manufacturing application at Section 3, they're going to find a lot more resistance than somebody that takes through a multivariate analysis. So you have to be sure that the value that machine learning process adds is necessary.
Thanks.
Okay.
Hi, thank you very much for your talk. I guess I represent one of the teams trying to apply machine learning to these tasks.
Okay,
so
excellent.
it was a remarkably similar question as the last gentleman, I'm
Yeah.
afraid. I was wondering which areas machine learning has been uptaken in so far and where you think the next one will be.
We've used it for, you know, looking at large particle data sets and things like that. So we've used it. It's very good for that sort of thing. You know, support vector regression and all sorts of things. It's taking it to that next stage where it's on the critical path for that. So whenever you've got a large data set and the tools to do it, you know, people use it for crystal structure prediction. People use it for things like that. The computing power is now there. I'm told by the CSP people that the graphics card in a gaming computer is you know, what would have been called a supercomputer. So if you parallelize half a dozen high-end gaming graphic cards, you've got computing power. The question would be, is the outcome transparent and would it meet expectations? You may well come to an answer. When you're trying to do crystal structure prediction, you're just trying to get a feel for whether you've got the right polymorph. But you're not then saying, I don't know if anybody's taken that to the regulators and said, we have the most stable polymorph. So it's that leap between coming up with an answer which is scientifically rational to being able to justify that in a regulatory environment.